Imagine you could build a tiny, custom machine, one that's designed to fix a problem inside a single human cell. Well, that's pretty much the challenge of designing new proteins. These molecules, they're the workhorses of biology, and figuring out how to create new ones from scratch, that is one of the biggest keys to unlocking a whole new generation of medicines. And, just recently, a new AI tool has made a really stunning breakthrough in doing exactly that.
So what if I told you that a new AI tool could design these custom proteins with a success rate as high as 73%? Look, in a field where getting into the single digits is considered a win, a number like this, it's a total game changer. It is a massive leap forward. But, of course, it also brings up a huge question.
Right? How can an AI possibly predict which of these incredibly complex little molecular machines will actually work in the real world? In this explainer, we're going to break down the tech that's making this possible, and it's all based on a new technical report from the team at ByteDance Seed.
Okay, so the hero of our story is a system called PXDesign. You should think of it as a complete end-to-end assembly line for creating brand new proteins. These are called binders, and they're designed to stick to a very specific target molecule, kind of like a key fitting a lock. And as the research shows, it's just way more accurate than anything we've seen before. So how does it pull this off? Well, the real genius of PXDesign is in its simple two-stage blueprint. It's actually a classic strategy. Go wide first, and then go deep. First up, you have the generation stage. This is where the AI just comes up with thousands and thousands of possible designs. Then comes the really critical second step, filtering. This is where a whole different set of AI models sift through all those possibilities to find the ones that have the absolute best chance of actually succeeding. Let me give you an analogy. The generation stage, it's like a massive, no-holds-barred creative brainstorm. The goal here isn't to find the one perfect answer right away. No, it's to flood the room with as many different and creative ideas as you can possibly think of. And then, the filtering stage brings in the expert critic. Its job is to go through every single one of those ideas from the brainstorm and pick out that handful of true gems. The designs that aren't just cool on paper, but are structurally sound and have the highest probability of working in a real lab.
And this is where we find the first really big innovation. See, to run that massive brainstorm, PXDesign doesn't just use one method. No, it's actually developed two completely different but complementary paths to create these molecules.
So the system uses two approaches. The first one is a diffusion model. You can think of it like an artist who has studied millions of paintings. It's built for speed, and its job is to generate a vast and diverse portfolio of options. The second approach, a hallucination model, is more like a sculptor, meticulously refining a single block of marble to optimize a design for one very specific goal. One gives you breadth, the other gives you depth. It's brilliant.
Now, the team really focused on making that diffusion model, which they call PXDesign-D, incredibly efficient. A key innovation here is its architecture. Unlike some of the older methods, PXDesign-D directly generates the 3D coordinates for every single atom. This one change lets it dodge these major computational bottlenecks, and the result is a massive, several-fold speed-up, especially when you're dealing with big, complex proteins.
And that speed? It has a huge real-world impact. I mean, the researchers themselves say that in just a single day, their diffusion model could produce more viable candidates than any of the slower, more targeted methods. When you're talking about drug discovery, that kind of speed is, well, it's everything. But, generating thousands of ideas is only half the battle. You still have to find the winners. And this brings us to PXDesign's second major innovation. Its incredibly clever approach to filtering. So here's the clever part. Instead of relying on a single AI critic, PXDesign uses multiple models to evaluate the designs, including its own model called Pretendix, plus models based on AlphaFold. And the researchers discovered something fascinating. These different models have different biases, like experts with different opinions. The winners that Pretendix picked had, and I'm quoting here, a surprisingly small overlap with the winners that AlphaFold picked. So by using them together, they just cover way more ground and catch more potential successes. It's honestly the perfect analogy. If you want to make sure your work is absolutely flawless, you don't just ask one person to review it, right? You ask two different experts. They're each going to bring a unique perspective, they'll catch different kinds of errors, and The final product is just going to be exponentially better. That is the core idea behind PXDesign's filtering system. Okay, so all of this theory sounds fantastic, but does it actually work? This is always the moment of truth for any AI system in science. That jump from the clean, perfect world of a computer simulation to the messy reality of a wet lab.
And wow, the results are stunning. This chart shows the experimental hit rates, basically the percentage of AI-designed proteins that actually worked in the lab. For the target PDL1, which is a key protein in cancer therapy, PXDesign hit a 72.7% success rate. Now compare that to a previous state-of-the-art method, AlphaProteo. It scored just 11.4% on the same target. That is a six-fold improvement. Just incredible.
So what does success actually look like down at the molecular level? Well, this is it. This is one of the AI's designs, created entirely from scratch, or de novo, perfectly docking with the exact target it was designed for. This is that key fitting the lock idea right here in action. And what's so crucial is that the AI isn't just finding one type of solution over and over again. It's generating binders with wildly different shapes and structures. And that's critical, because it's not just about finding a key that fits the lock. It's about exploring thousands of different key designs to find the absolute best one.
And you have to remember, every single one of these successful designs isn't just an academic victory. No, it's a potential new tool. It could be a new diagnostic, a new therapeutic, or maybe even a brand new key to unlock some fundamental biological puzzle we haven't solved yet.
So the big takeaway here is that this whole innovative approach, the two-stage generation and the smart filtering, it isn't just a theoretical improvement. It translates directly into a dramatically higher success rate where it really counts, in a real-world lab producing a diverse portfolio of molecules that actually work.
This breakthrough in designing proteins is absolutely incredible, but honestly, it's just the beginning. The underlying framework that makes PXDesign work, well, it opens the door to a much, much grander vision.
Because the core architecture that powers PXDesign isn't limited to just proteins, the next frontier is to use this as a unified model that can design a whole range of critical biological molecules, from things that bind to DNA and RNA all the way to brand new enzymes.
I mean, the researchers have already shown that the same core engine can be used for designing cyclic peptides, for creating molecules that bind to DNA and RNA, and even for designing small molecule drugs. This really transforms it from just a protein designer into a true unified molecular design platform.
And that leaves us with one last really tantalizing question. The ability to design proteins with 73% accuracy is a revolution all by itself. But what happens when that power extends to almost any key molecule in biology? What new medicines, what new materials, and what new technologies will we invent when we can finally design the molecules of tomorrow on demand?